Right. So the first step of photosynthesis, which is the light-dependent reaction, it occurs in the thylakoid of the chloroplast, and it is all entirely controlled by these two photosystems, the photosystem one and photosystem two. Whereas this like photosystem is not like something special. It's actually that reaction center that was discussed the previous slide, like which was the antenna chlorophyll when they pass the energy directly to the reaction center, which is usually the chlorophyll A. Now that chlorophyll A will now help in the entire process of the light reaction by transferring the electron, the excited electron, to the adjacent electron acceptor, and so on and so forth. The electron, the energy will get passed down to the like different molecules via an electron transport chain, and that will lead to production of ATP and NADPH. Once this is produced, this ATP and NADPH will now enter into the stroma region, means it will just simply move out of the chloroplast, the thylakoid of the chloroplast, and then will straight away initiate the process of the Kelvin cycle, which is responsible for production of the sugar. Now, to know about it slightly in detail, then we can say that the light energy is coming and hitting this center, this like the thylakoid. This is actually the thylakoid membrane, and in the thylakoid membrane we can see right there that the photosystem 2 and the photosystem 1 is present right there. And what is happening there is that the photosystem 2 is carrying that particular like signal, and then it is passing straight away one after another, and the electron is getting like the primary electron acceptor is actually right there seated, where it is just taking that energy and will transmit it later on to the next molecule. So the prime this actually the first set that we are seeing right here, right there, they are actually the antenna chlorophyll. They are passing the entire signal to this one. This is the reaction center. So this is the one, this entire unit is behaving as a photosystem 2 definitely, but the photosystem 2 is made up of multiple chlorophyll, you can say. Like there are numerous chlorophylls are there present, and the reaction center at at the dead center, and with the reaction center only the electron acceptor will be there present, and by that electron acceptor that the electron it actually gets passed down to the next. What actually happen is in the molecule itself, upon excitation by the light energy, a pair of electron is there, which is a tendency to jump out into the higher orbit that is normal, orbit that we can say it's present. And while jumping up to the normal higher orbit, it actually creates a resonance effect of transfer of energy, because whenever the electron jumps into a higher orbit, it has a tendency of coming back to the previous normal state, the ground state orbit. But to come back, it has to lose the energy, the excess of energy that it has gained. So this is usually by means of either production of light, or by emission of light will take place for that, or it is like some heat energy will be generated. And here in this case, since that is not happening all the time, because that transfer of energy will not take place like that way, though the resonance effect is there that the energy will get transferred to the next molecule, chlorophyll and all. They are actually showing that resonance transfer of energy, but what is happening in this reaction center is, as the electron acceptor is sitting right next to it, so the electron acceptor will immediately take this higher state electron, because the electron moving up into a higher orbit has a tendency of slightly moving away from the entire atom, you can say. So I mean the molecule itself. So when that happens, the electron acceptor being positioned right next to it, it has a tendency of capturing that electron, that free electron, and then passing it to the next. So that way the transfer of the energy is taking place from one molecule to the next. Yes, there is one more step that is going on right here, because it is not that the chlorophyll is actually always producing the energy, I mean the electron. The electron is also coming from water, because water is also being hydrolyzed, not hydrolyzed, sorry. water is actually being split right there, called the photolysis of water, because there is an enzyme sitting right there which, due to this excitation of the chlorophyll, has a tendency of splitting water molecule into protons and oxygen. And this is the step where the oxygen is actually generated. You can see it's shown right there. This is the step where oxygen is actually generated. So once this oxygen is generated, this oxygen will simply diffuse out of the membrane, and like all the membranes, it will just simply diffuse out of it, and that will be the oxygen released during the process of photosynthesis. Now, this transfer of this energy that we are seeing here, it actually, this electron that is being transferred to the, like, electron acceptor that is present right next to it, is the one which, like, pass it on to through a series of electron acceptors. And then there is another photosystem sitting right there, with which is called the photosystem 1. Once it reach that particular photosystem, there is actually like a process of chemosynthesis, like chemiosmosis, we call it. This process, entire process of chemiosmosis is there. So that chemiosmotic process will take place, where the conversion of NADP into NADPH is going to take place. So let's learn about that entire system in detail. So this is the entire model that is present there. You can see there are this photosystem 2 and the photosystem 1 shown right here. So as told, as said before, that photosystem is nothing but that chlorophyll molecules only. So keep in mind, here there are actually two numbers are written there, the P680 and the P700. It's written right there. Okay, so this word, it actually means P, it stands for the pigment. 680, the number, it actually represent the wavelength of light it accept. Okay, means at 680 wavelength of light, which is in the, like, reddish zone, means it's actually orange to red like color. So that particular wavelength of light is something that the photosystem, means that chlorophyll A is actually able to accept, and is going to use that light energy to release that electron, because gaining that particular wavelength of light, the electron will be released, and finally that electron will pass on to this electron acceptor, which is, as I mentioned before, that it is going to be present or positioned right next to the photosystem. So it is going to be accepted by this particular photo, means this electron acceptor. This is called plastoquinone. You can read the name right here, plastoquinone. And not only that it is accepting the electron and transferring it, but along with it, the proton released out in this process is actually like the proton also is also getting like transferred through it, because you can see it is accepting proton and electron all together, and thus is getting reduced, PQH2. It is the reduced state of the plastoquinone. But that reduced state will not stay as it is. So what will happen is that it will transfer that proton down below into the thylakoid lumen, the gap, the chamber in the inner portion of the thylakoid. So it is going to transfer it down into the thylakoid lumen, and at the same time, the electron is going to get transferred to the adjacent electron acceptor, which is cytochrome B6F. Okay, this molecule is called cytochrome B6F. So this cytochrome B6F is the next electron acceptor. So what it will do is it will transfer the electron to the plastocyanin. Now plastocyanin is a, like, transporter, even you can say. It's like taking this electron and it will move straight to this photosystem 1, which is of, the number is written as P700, means it absorbed the 700 wavelength of light, red. So this particular molecule, now again taking the light energy, will now get excited and transfer the electron to this ferredoxin, another electron acceptor which is attached with it. And then this ferredoxin will be, like, present. It actually has this molecule, this actually is a enzyme right there. It is called ferredoxin NADP reductase. The name is written right there, ferredoxin NADP reductase. So this NADP reductase, it has one purpose only: reduce the NADP into NADPH2. For that, again, it will take a large number of protons. And it is not only the proton, the electron is transferred from here, and that way it is getting reduced to NADPH, or NADPH2 also it is written in many of the places. Same thing. Now, keep in mind, there's one fact I have to share here. It is that the name is written as PS1, means photosystem 1 here and photosystem 2 here. It's actually because of like the discovery. This particular photosystem was discovered earlier than this one. I'm sorry, than this one. That's why this is named as 1, and then this is named as 2, because usually you'll think it's otherwise, that the first one should be the named as 1 and the second one should be named as 2, but it is actually due to the discovery of the of this one is actually it took place earlier. That's why it's named such a way. Now, once this process actually end in at the form of this production of NADPH, now what is actually going to happen is we may think that the process is over because the electron transfer is as completed. It ended right there, isn't it, with the production of NADPH2. But at the same time, there are a large number of proton which is present in the lumen, isn't it? There are a large number of proton that are present in the lumen. This proton will actually be pumped out with the help of this particular molecule. This is another enzyme. This is called the it's a proton pump. Simply speaking, it is a proton pump, but it goes by the name of ATP synthase, and you are familiar with it by the name of oxysome. Oxysome that can be found in the membrane of the mitochondria, and it is responsible for formation of ATP. So what it does is, it is actually the same thing. It's actually, its purpose is to push the proton out from the lumen into the chloroplast stroma, means the matrix of the chloroplast. And while moving out at that time, it has like the property of production of it has a property of production of ATP by joining an ADP and a phosphate together. So this is a process which is like phosphorylation. We can say it's a process called phosphorylation because it is taking place where adenosine diphosphate is being phosphorylated, and since it is connected with a process which is dependent on the light, so this particular step is sometimes called a photophosphorylation. So you have to keep in mind the entire chemiosmotic model is actually ending with the photophosphorylation step. So we can say that it begins with excitation of the chlorophyll, photolysis of water, the entire electron transport system leading to reduction of NADPH, I mean reduction of NADP into NADPH, and finally production of ATP by photophosphorylation. These are the steps of the light dependent reaction of photosynthesis.